Hello, everyone. I hope you still have some energy. So this is Heba, originally from Egypt, but now I'm based in Sweden. And my research is focusing on understanding estrogen receptor-mediated neuroprotection using mouse model of Alzheimer's disease. So let's start first by what is Alzheimer's disease. Alzheimer, if you may know or have heard about it, it's a neurodegenerative disease characterized by memory deterioration as one of the hallmark, and also nerve deterioration. So the nerve deterioration caused by abnormal accumulation of protein. This protein is normally produced by the body, but for some reasons it is accumulated in the brain and it kills the nerve cells. The problem here is that it accounts for 70 to 80% of people who are living now experiences Alzheimer's disease, and this number is expected to triple by 2050 to be more than millions. And the other problems that is most of the reasons or the causes are sporadic without a known reason, but only 5 to 10%, which is due to genetic mutation, non-genetic mutation. So we are working to understand what is the reason behind the sporadic AD forms, and one of them is age, which we can't stop. So if we look here, if you like doing the math, 1 in 9 people who are living now experienced Alzheimer's disease, and they are more than 65, and this number is expected to be 1 in 4 by 2050. With the age, our hormone level changes. So if we look here on the right side, you can find that there is the blue one is for testosterone, which is the male androgen hormone, and the purple one or the lavender one is the hormone estrogen for female. And with the time, this hormone gradually decline in male, while it is in female, the red one is sharp decline after menopause. And this is a big problem for women because this makes women have higher risk to develop Alzheimer's disease with age. So 2/3 of Alzheimer's disease patients are women, and this one related to the estrogen sharp decline. So they expected that the sharp decline of estrogen increase the vulnerability of women since estrogen itself is neuroprotective. So our role is to understand how estrogen is the neuroprotective and what happens when we lose our estrogen hormone level in the brain. And this is my research questions. And when I looked in the literature, you can find me here crying. What is happening on? Exactly, this is the case. If we said that after menopause, we can give just hormone replacement therapy to delay the onset of Alzheimer's disease in women, this is what I have found. A lot of debate. Hormone replacement therapy is good. Hormone replacement therapy is terrible. What we believe, we don't know. Till now, it is still debating. So we need to understand how estrogen itself works. So if we understand the mechanisms behind the neuroprotection, we can just develop something during the pathway of estrogen to delay the onset of Alzheimer's disease in both male and female, and also to help treating Alzheimer's disease. So our research here, using a mouse model of which increase amyloid production. So amyloid beta is the protein that I have mentioned in the beginning. It is the one that accumulate in the brain and kills the nerve cells. So we have a model of Alzheimer's disease called ABB-NAGF, with genetic mutations that lead to accumulation of amyloid beta. And this model we have knocked out of one of the estrogen receptors. Thanks for the first talk who covered estrogen, and it is estrogen receptor, so you know exactly what I'm talking about. So estrogen receptor beta, we focused on first because estrogen receptor alpha is more related to reproductive function, while estrogen receptor beta is thought to be neuroprotective that has memory-related function in the brain. So first we started by targeting this one, but we aim also to target a second receptor later. So the first thing, I'm a wet lab person, so I started as a wet lab and I ended as a bioinformatician. I'm just fighting inside myself now what I want to be, so we will know later. So we started with assessing the behavior of the animal, it's the ABB-NAGF and the one without the receptor. And after tissue processing and data analysis, surprisingly, we find that when we knocked out for this estrogen receptor beta, I found the one in the left side, the green one, if you see colors. So this is the green one. You can just here. It's the male. So when we knocked out for estrogen receptor beta, we found that males, but not female, have higher risk to develop amyloid beta more than females. And we have confirmed this with measuring amyloid beta level itself by ELISA, PCR, and also other functional analysis. And when we find all the results are more related to the hippocampus, which is the region in the brain that's responsible for the memory, we sequence it for this hippocampus region in the groups that we have, the wild type or the one with the receptor and the one with the knockout of the receptor. And we got this hippocampal cell population. The major cell population is the neurons for sure, and also they have microglia, which is the cell or the resident immune cells of the brain, the defense mechanism. We have the astrocytes, which is supporting cells, and we have also other cell types. Interestingly here, what we got is that male at higher risk in our model. This is still ongoing, and to just know what is going on, we have heard these two mice listening to us in the session, and unfortunately, Alzheimer's disease never happens in mouse. So there is no sense of what we are doing if we can't translate our research into human. So this is a coming question of what we get out from the mouse. We should translate it by using either post-mortem brain samples, do the same following the hints that we get from the sequencing, or whatever, using published human data and analyze and follow the targets. We have interesting targets for now, and we are following until my PhD. And if you're interested to know more about estrogen receptor beta, please follow the mice and come to my poster today or in the main session of the conference B06. And this is like a screenshot of the poster if you are like, can't find your way. Thank you. This is my group, and also this is